White rust is an important water convolvulus disease in Taiwan. It is prone to occur in summer season. In order to prevent the occurrence of disease, a new disease-tolerant line, namely Fengshan 95455, has been selected. This new line performs fine texture, needle leaf shape, numerous tillers, and does not brown after cooking. It is suitable for ratoon culture. In order to fit the requirement of more diversified and new tasty vegetable crops, a new breeding line of purple cauliflower, namely A20, has been selected. This new line performs early mature. The flower curd can be harvested at 60 days after transplanting. The curd is brilliant in purple-red color, compact, fine texture, and good taste. Cultural practice improvement of horticultural crops. Development of net house culture system. Net house culture efficiently prevents the occurrence of papaya ring spot virus disease, damage of fruit, and melon fly of carambola and Indian jujube. It not only increases yield, but also improves quality. There are about 90% of papayas, 80% of Indian jujube, and 40% of carambola that are cultivated by means of net house culture in Taiwan. Development of wax apple forcing culture techniques. The normal wax apple production period in Taiwan is from May to July. The fruit quality is poor due to high temperature, high moisture, prone to crack, and damage by fruit flies. In order to produce high-quality wax apple, the branch has developed forcing culture techniques. The vegetative growth of the plant is inhibited, and the reproductive phase is enhanced after treatment by means of waterlogging, root pruning, girding, and shading by black plastic net. The fruit production period can be shifted from November to the following year of January. It can also be from 1 year 1 harvest to 1 year 3 to 5 harvests. Nowadays, it has become a very important cash tropical fruit in Taiwan. The fruit derived from this technique performs uniform, brilliant red, high sugar content, and excellent qualities, and thus is quite welcome by producers and consumers. Development of banding culture of guava and lodging culture of papaya. This branch has developed banding culture technique for guava year-round production. Papaya lodging culture is not only dwarfing plant type, but also increased the efficiency of net house space using rate. It has also prolonged the papaya production period in Taiwan. Establishment of dwarf and close planting system of Yuhu Bao lychee. Yuhu Bao lychee is an excellent early mature variety. Due to low yield of this variety, it was not welcomed by the growers in the past. This branch has established a dwarf and close planting system for Yuhu Bao lychee production in 1996. This system is welcomed by the grower, and thus the planted acreage is strikingly increased to 3,000 hectares in last 10 years. It has completely changed the structure of lychee industry and marketing system in Taiwan. Establishment of soilless culture system for leafy vegetable production. In order to overcome the problems of unstable weather condition, degradation of soil properties inside facilities, serious occurrence of pest disease, and disorder of successful cropping, a floating root hydroponic cultural system was successfully established for leafy vegetable production. Establishment of baby leafy vegetable production system. In order to stabilize the summer leafy vegetable supplement in the market, this branch has established the production system for baby leafy vegetable crops. The baby leafy vegetables perform fast in growth rate, and it's unnecessary to spray any pesticide during growth period. The bok choy, green stem bok choy, Chinese kale, and amaranth are considered to be favored for baby leafy vegetable production. The DNA gene pool of many tropical fruit trees such as lychee, mango, and papaya have been established. This technique is applied for identification of genetic source, evaluation of parental relationship, shortening of breeding period, and evidence of breeder's right of new developed tropical fruit tree varieties. This branch has developed a DNA marker by RFLP technique for identification of sexual expression in papaya seedling stage. The accuracy rate could reach 98.8%. For rescuing interspecific hybridization embryo in brassica vegetables. The barriers of outcross breeding in brassica vegetables have been overcome by plant tissue culture via embryo rescue technique. The interspecific hybrid plants such as cauliflower bok choy, cauliflower mustard, and Chinese kale rape, and intergeneric hybrid plants of Chinese kale radish have been developed. This technique consequently broadens the genetic variation and improves the selection efficiency in breeding. In order to improve the efficiency on detecting and identifying plant pests and pathogens, this branch has successfully developed ELISA techniques for identifying the Anthurium bacterial blight, lettuce, and Cucurbitaceae crop viruses. Meanwhile, rapid diagnosis of bacterial diseases was also achieved using PCR techniques developed by the branch. In order to prevent the loss of genetic resources of tropical fruit trees, this branch has established a National Tropical Fruit Tree Genetic Resources Conservation Garden in 1994. There are 44 families, 155 species, and 261 varieties of tropical fruit trees that are conserved. In order to diversify the tropical fruit in the market, this branch continuously collects and introduces new tropical fruit trees for the observation of adaptability and for the regional trail. The pitaya, red and yellow rambutan, jackfruit, cempasúchil, and abio are recognized with great potential in the future. This branch provides free diagnosis service on plant diseases and insect pests for farmers, schools, and institutions in southern Taiwan. Farmers are notified of the results of diagnosis and suggestion for dealing with these problems by email or by phone. According to the record, there is a yearly average of over 200 cases on plant disease and pest diagnosis service in this branch. The annual control calendars of diseases and pests on 5 tropical fruit trees, including mango, wax apple, guava, Indian jujube, and pineapple, were set up and offered the information to the farmers on pest and disease control. In addition, pineapple wilt was a destructive disease in Taiwan during 1940. After years of study, researchers confirmed that pathogen was transmitted by mealybug and proposed integrated strategies for controlling the disease. The occurrence of malformation of fruits in papaya, mango, and Indian jujube was confirmed due to boron deficiency in low temperature and drought season. These problems could be solved by applying boron in soil or spraying boron solution on fruit trees during proper period of fruit production. In order to reduce the application of pesticides, bagging methods are investigated to prevent the infection and damage of pathogens and pests on mango, lychee, carambola, wax apple, sponge gourd, and bitter gourd in recent years. Studies on non-pesticide control and monitoring of insect pests on Indian jujube, carambola, and papaya were conducted, such as controlling citrus red spider mite by predator mite in greenhouse, monitoring the population of oriental fruit fly, and small yellow thrips by yellow and blue sticky papers, monitoring the annual population fluctuation of orient fruit fly by yellow sticky paper in orchards. Meanwhile, efficacy of oils and saponins on controlling spider mites, thrips, and mealybug were also evaluated. Recent researches on plant fungal diseases are focusing on ecological surveillance of pathogens, as well as establishing tactics for controlling diseases on tropical fruit trees. In these studies, the causal agent of stem canker on guava has been confirmed, and methods for controlling this disease have also been suggested. In addition, researches on screening fungicides for controlling guava wilt pathogen were carried out. A semi-selective medium for detection of guava wilt pathogen was developed, and studies on the ecology of the pathogen in the field is undergoing. On the researches of bacterial diseases, Anthurium is an important floral plant in Taiwan. This branch is not only assisting the diagnosis of bacterial diseases on Anthurium, but also offers serums to other research stations to proceed seedling health inspection projects. Meanwhile, recent researches were focusing on collecting strains of black rot pathogen from Basetia plants, as well as investigating the characteristics of these pathogens in Taiwan. Breeding projects on screening resistant Brassia varieties to black rot disease were also undergoing. In order to increase the additive value of horticultural crop products, this branch is focused on research of aromatic herbs utilization, functional extracts of mushroom, and rice fermentation techniques. The complex essential oil extracted from aromatic herbs is embedded to fiber for insect-repellent textiles. The polysaccharide is extracted from the fruiting body of mushrooms. It is applied to cosmetic moisturizer, intestine functional improvement food, and biodegradable biomaterial. The technique of rice milk fermentation for replacement of milk medium in lactic acid fermentation has been developed. This processed product of rice lactic beverage, grass doo-doo, is made. Selection of proper cover grass for slope land orchard management. Favorable for crop growth and sustainable for better soil fertility in slope land orchard and vegetable farm are rather important. This branch has set up a soil and water conservation outdoor classroom and demonstration farm for teaching school students, farmers, and citizens. This branch has established an efficient irrigation and fertilization system for saving energy and reasonable nutrition management of slope land orchard. In order to sustain crop culture, increase competition ability, and connect to international market, this branch will focus on improvement of tropical fruit trees and vegetable crops breeding by biotechniques, establishment of safe production and virus-free seedling propagation system, improvement of forcing culture technique, development of organic energy-saving and lowering carbon emission culture system. Thank you for your visit. Your comments are welcomed.